So it's a gram-negative rod and it is motile by unipolar or monotrichous flagellum. Alright, again it is cop or policeman organism in the sense that it is catalase and oxidase positive. COP. Catalase and oxidase positive it is. It is a non-lactose fermenter. Now, coming to the diseases by this organism. Remember, it is the most common cause, MCC, most common cause of keratitis in contact lens wearers. Contact lens wearers. If somebody asks you what is the most common protozoan causing keratitis in contact lens wearers, then your answer is going to be acanthamoeba, which I have discussed separately in acanthamoeba module. Alright. Then secondly, it is the MCC most common cause of burn wound infections. When the skin integrity is lost, as in case of burns, the wound becomes susceptible to infections, and Pseudomonas aeruginosa is notorious to infect these burn wounds. Then this is Pseudomonas infection of the burn wound. Remember, the first point which I want you all to remember by just knowing its name, Strongyloides stercoralis, is it is strong enough to penetrate the skin. So the mode of infection in this case is again skin penetration, just like hookworms. Okay, so remember the mode of infection. The mode of infection is skin penetration, strong enough to directly attack the skin, penetrate the skin. Then the infective form, remember a while back what did I tell? Who is going to penetrate the skin? Is it the egg or is it the larva? Of course not the egg, because egg is supposed to be ingested or eaten. So the infective form over here is again filariform larva. Filariform larva. Now guys, what will happen when filariform larva penetrates the skin? Let's say it penetrates the skin of an individual. Now the story is going to be in the beginning similar to what we saw just now in case of hookworms. That is, it is going to enter the venous circulation. It will reach the right side of the heart. It will exit the pulmonary artery. It will then pierce the pulmonary capillary. It will gain access to the lung parenchyma. Alright, then it is going to ascend up the lungs and it's gonna reach the throat. Then it is going to be swallowed and it reaches the SI, that is small intestine. SI is my own short form. Okay, please don't use this in your theory exams in college. You write full form only, small intestine. Okay, so this reaches the small intestine. Till now the story is same. Then what is the difference between hookworm and strongworm and this thing, Strongyloides? As I said, during all this travel, morphological changes are going to happen in the larva. So in this case also, in case of Strongyloides also, by the time the larva reaches the small intestine, it is potent enough or mature enough or competent enough to transform into adults. So adults form, that is male and female. Now guys, here comes the twist in the tail. Over here, the female worm is not oviparous, instead it is ovoviviparous. Okay, and this is the only worm in the entire helminthology, which is ovoviviparous. The eggs are going to immediately hatch into the larva, and this larva is what? Rhabditiform larva. So we have got rhabditiform larva in the intestine. Rhabditiform larva can change into the infective form, that is filariform larva, in the intestine itself. So you only think that when infective form, that is filariform larva, when it is being formed in the intestine itself, can't it initiate the cycle again? In other words, can't it initiate auto infection in this person? This filariform larva can again penetrate the gut of the penetrate the gut wall, manage to enter the venous circulation, and the entire story will be repeated. So this in this case, what we are going, this is called as what? When the infective form is being formed in the intestine itself, it has the potential to initiate the cycle once again. So auto infection can occur. All right. And if somebody's immunity is very much jeopardized, what will happen? The immune system cannot take up, the immune system might become overwhelmed because of filariform larva present in the gut. So the filariform larva might not be taken care of by the immune response in case of an immunocompromised person. So auto-infection is more likely to occur in such people whose immunity is compromised. Okay. After sexual intercourse, there's an incubation period which ranges between 9 to 90 days, after which a patient who develops syphilis, if we look at the untreated, if we look at the natural history of disease in untreated cases, what we are going to see? We are going to see that the patient will go through certain stages of syphilis, of which the very first stage is known as primary syphilis. Now, primary syphilis is characterized by a painless, hard to touch or palpate, that is indurated, hard or indurated to touch or palpate, genital ulcer known as, K/A is known as hard chancre or Hunterian chancre. Alright, now it is called as hard chancre because it is hard to touch or palpate, and it is called as Hunterian chancre, named after Sir John Hunter, who actually distinguished gonorrhea from syphilis. Okay, remember, gonorrhea is also a sexually transmitted infection, but gonorrhea comes under the category of discharge causing sexually transmitted infections, whereas syphilis is it comes under the category of ulcer causing STIs. With painless, hard, indurated genital ulcer, there is going to be also painless inguinal lymphadenopathy. Okay, painless inguinal lymphadenopathy. Alright, and remember, these lymph nodes and the genital ulcer, these are highly infectious lesions. Okay, these are highly infectious lesions. Microscopy and serology. Now guys, it makes sense to do microscopy in the earlier stages of the disease, that is in primary and secondary stages, because the shorter the duration of infection, the less likely it is less likely that antibodies would have been produced, and it is more likely that organism will be could be encountered in the clinical specimens. So it makes more sense to do microscopy in the initial stages, whereas serology, wherein we detect antibody in the patient serum. Okay, antibody detection is done in as far as syphilis serology is concerned. Serology is more, it makes more sense to do serology in secondary and tertiary stages of the disease. Okay, because in the later stages, when suppose the secondary stage is almost towards the end and tertiary stage is about to start, we don't expect the organisms to be encountered in the specimens that commonly, whereas antibodies can be expected, right? So as far as microscopy is concerned, number one, as we know, dark ground microscopy can be done, but again, dark ground microscope is not available everywhere, and it has got that is one problem with it. And other disadvantages of dark ground microscopy is that the specimen has to be seen, the specimen has to be examined, or the smear has to be examined immediately, otherwise the organisms will die. Okay, smear has to be examined immediately. what we are on a lookout when we use dark ground microscopy to visualize the sample of the patient, we have to see the motility. Okay, we have to see the motility. So you have to see this the smear which you have made from the specimen immediately to comment on the motility, right? This is one problem. The other problem with dark ground microscopy is that it cannot differentiate between pathogenic and non-pathogenic treponemes. All right, it's very difficult to differentiate between pathogenic and non-pathogenic treponemes just on the basis of motility. So it is not commonly done. Then we have got direct fluorescent antibody, that is DFA staining, direct fluorescent antibody, that is DFA stain for Treponema pallidum. A child presented with pharyngitis and a swab was obtained, pharyngeal swab was obtained, sent for culture. Swab should be discarded in which colored bin? Yellow, red, white, blue. Answer is, guys, yellow. Okay. The infected swabs, okay, like this, or cotton swabs, they go to yellow color. Okay. So straightforward question, nothing much to discuss over here. Okay, by the way, tell me, child presented with pharyngitis, swab was obtained. हमारा जब गला खराब होता है, do we really send our in India? Is this a routine practice of sending the throat swab for culture? Unfortunately, it is not done, but that is the norm actually which should be followed. Okay, and only then the antibiotics should be started if it is a bacterial infection. Otherwise, the most common cause of sore throat is what? Viruses. So IgM antibody detection can be done by a method, that is by a test, standard agglutination test. Standard agglutination test, which I already mentioned before, is pili for Gonococcus. Alright. Now these pili help the organism, to attach to the urethral and endocervical epithelium. Remember, this organism causes urethritis and endocervicitis. So attachment to urethral and endocervical epithelial cells is brought about by pili. That's why pili are such important virulence factors. In males, it will cause urethritis, which is characterized by urethral discharge and other features like burning, micturition, etc. Now, remember, males are usually symptomatic, and the classical complaint of a male is urethral discharge. Males, the best sample is of course urethral discharge. Sometimes frank discharge or very florid discharge may not be present. Okay, so if this is absent, the urethra can be milked and the discharge can be expressed. So if this is absent, milk the urethra and express discharge. If this also does not lead to any expression of discharge is not collected. So if this fails, then the last resort is collect a periurethral swab. Collect a periurethral swab. A female presented with profuse white vaginal discharge without any itching on 15th day of menstruation. What can be the reason? Physiological, candidiasis, trichomoniasis. Now the color is white, automatically it rules out trichomoniasis, which is characterized by a greenish. Remember GIF नहीं होता, actual full form तो नहीं मुझे पता, जो technical language में होता है GIF नहीं होती, जो greenish, itchy, frothy vaginal discharge in trichomoniasis, so that is automatically ruled out. D option I could not get. Okay, some people were thinking of white discharge, candidiasis because of the color, but guys, there is in candidiasis intense pruritus is a very debilitating feature. Intense pruritus. Okay, so but this female does not have any itching, and the buzzword, the clincher in this question is
The discharge was appreciated on 15th day of menstruation. Guys, answer is physiological. Okay, physiological.
The third category which I'm going to introduce to all of you is halogens. Now, halogens we have read in school in chemistry, and they have got different roles. What about their role in medical practice? Now, amongst all the halogens, you need to remember iodine and chlorine, which have antimicrobial activity or antimicrobial effect, and these halogens, they easily combine with the sodium and all, and calcium, etc., to form salts, and these salts can be used as chemical agents in the practice, in our endeavors to achieve sterilization and disinfection. So sodium salts are very commonly used, most commonly used are used for antisepsis and disinfection. Now, between iodine and chlorine, I'm going to first talk about the chlorinated compounds. Chlorinated compounds. Now, the very first fact which I want you all to remember regarding chlorine or chlorinated compounds is, remember, C for chlorinated and C for corrosive. So, the very first point which you need to remember about these compounds is that they are corrosive. So, they are not used as antiseptic agents. They are not used as antiseptics. Okay, first point. So, they are used only as disinfectants, and they're not going to kill everything, so they are not sterilants. Okay, so they are used for the purpose of D, that is disinfection, and not sterilization, and also not for antisepsis. Now, examples of such chlorinated compounds which you need to remember are sodium hypochlorite, that is famously known as household bleach. 5.25% is the concentration used most commonly. Then we have got calcium hypochlorite, that is bleaching powder. Then chlorine gas. Alright, so these are the important compounds which are derived from chlorine, and no matter which compound it is, they all have the same mechanism of action, guys. And what is that? The MOA, that is the mechanism of action of all the chlorinated compounds, is that they are very good. They are very good oxidizing agents. All of these compounds release what is known as hypochlorous acid, and the oxidizing capability of these compounds is by virtue of this acid only, that is hypochlorous acid, HOCl. Okay, so because they are very good oxidizing agents, what they are going to do, they are going to carry out oxidation of cellular material and destroy most of the vegetative bacteria and fungi. Then the next thing which you need to remember is they have got good activity or good action on. They have got good action on, remember, G plus bacteria, G minus bacteria and viruses. Okay. Now guys, there's a reason why I have chosen only chlorinated compounds for discussion over here is, remember a few minutes back I was talking about phenol, where I told that it has got good activity on GGM, that is G plus bacteria, G minus bacteria and Mycobacterium tuberculosis. Now over here, as far as the good action is concerned, I have just replaced MTB with viruses. So over here you have to remember GGV and not GGM. Okay. Now this point has got a very important significance. You know why? In the discussion about phenol, I told you to n-box or encircle MTB, and over here I would like you all to n-box or encircle viruses. Now because chlorinated compounds have got such good activity on viruses, therefore These are the agents of choice for managing blood spill. And what agent we use? We use most commonly amongst all the chlorinated compounds. It is sodium hypochlorite. So write down, sodium hypochlorite is the agent of choice, chemical agent of choice to manage blood spill. Now, what is the relation between blood spill and viruses? What are we, most scared of or afraid about whenever an accident of blood spill happens? We are actually afraid of or scared of blood-borne viruses like human immunodeficiency virus, hepatitis B virus, hepatitis C virus. And because chlorine has got such good activity on these viruses, that is why it is by far the most, the excellent, the best method, the best chemical agent of choice to manage blood spill. And for this purpose, we use what concentration? 1% sodium hypochlorite is used to manage blood spill. So this is very, very important. And next, you need to remember it has got poor action on, the chlorinated compounds have got poor action on, remember, spores and Mycobacterium tuberculosis. So in phenol, we remembered PSV, that is poor action on spores and viruses, and over here remember PS and M, that is poor action on spores and MTB. So as you have seen, chlorine also is not going to, kill everything. So obviously it is not going to carry out what? Sterilization. And already I have said that because it is corrosive, you cannot use it on human tissue. So it is not going to be used for the purpose of antisepsis. These are mere disinfectants. Then just like phenol, the activity of chlorine decreases in the presence of organic matter. So you need to use a higher concentration of the agent which you are using if suppose you are dealing with organic matter, okay, like blood, feces, etc. Then next what you need to remember about these compounds is that sodium hypochlorite, which is used most commonly, it is unstable, it disintegrates easily, therefore a fresh solution is to be made daily. This is very, very important. Plus, chlorine reacts with organic to form trihalomethanes, which are carcinogenic. So this is very, very important to remember. So guys, this is about the most important points you need to remember about the chlorinated compounds, which we are discussing under the category of halogens. Remember, we are doing chemical agents category number 3. Halogens, I told you, have got chlorine and iodine, and we are done with chlorine now. Now, if we talk about diagnosis of bacillary dysentery, the sample of course which we are going to collect are the feces, and as I discussed in detail how a fecal sample should be processed while I was discussing about the lab diagnosis of enteric fever, same principle guys is going to apply over here also. That is, we have to remove away the traffic of commensal organisms in the feces. How are we going to do that? You have to inoculate the fecal sample in the enrichment broth first. Enrichment broth, that is, you can use selenite F broth. Remember, F stands for feces. Or you can go for gram-negative broth. Okay. Now make a box and note down. Tetrathionate broth is not used if you are suspecting Shigella. Tetrathionate broth is not used for Shigella because it is toxic to Shigella. Okay, so if you are suspecting that the patient might be having infection with Shigella, do not inoculate the fecal sample into tetrathionate broth. Go for either selenite F or gram-negative broth. right? The idea of enrichment broth is that it will tone down or decrease the number of commensals and pathogens. If at all any are present, they will multiply. Okay, and you have to incubate the fecal sample in the enrichment broth for 6 to 8 hours. Incubate for 6 to 8 hours. Okay, and then do subculture on selective media. On selective media. Remember, S for selective, S for solid. Selective media are always solid. Tricho, the name itself has got tri. Okay, so trichophyton is going to infect all the three, that is skin, hair and nails. Okay, skin, hair and nails. We have got hair perforation test. Now, in this test, we look for the formation of wedge-shaped perforation in the hairs we have got hair specimen with us in the lab, and we mix it with the suspected fungus, and we see whether any wedge-shaped perforation forms or not. If the perforation is formed, then it is interpreted as a positive test. Alright. So, this test is positive and negative in respectively. Again, trichophyton mentagrophytes and trichophyton rubrum may it is negative. Alright. So, both these tests, the urease test as well as hair perforation test, both are positive in mentagrophytes and negative in rubrum. So, just for a quick recall, paramyxoviruses, of course, these are RNA viruses. They have got negative sense RNA. Alright, negative sense RNA. They have got helical nucleocapsid, and when we say helical, by default they have to have an envelope E plus, because there are no naked helical viruses. Okay, so they have got an envelope. And what did I say? These myxoviruses, their envelope has got three kinds of papillomas, H, N and F. Alright. The onset of illness in the form of measles prodrome. The measles prodrome is characterized by, remember, fever plus 3 C's plus photophobia. The 3 C's include cough, coryza and conjunctivitis. Fever plus 3 C's plus photophobia. The 3 C's, cough, coryza and conjunctivitis. Because of conjunctivitis, the patient is going to have photophobia. Okay, so this is the measles prodrome. Alright, so after this, this febrile prodrome, where the patient is having such respiratory features along with the fever, there's going to be development of a rash, a maculopapular rash. Okay, remember, measles is best defined as a febrile illness of childhood characterized by rash, but before this rash also something else happens, and that is pathognomonic of measles, known as Koplik spots. Okay, so we defined the incubation period as 10 to 14 days in case of measles. So guys, just remember, day 10, day 12 and day 14. Day 10, day 12 and day 14. On day 10, fever occurs, and on day 12, Koplik spots, and day 14, rash. So Koplik spots appear a day or two before the onset of rash. Alright, and these Koplik spots, they are pathognomonic. They are pathognomonic of measles. And what exactly are these? The Koplik spots. Koplik spots, guys, these are bright red, that is erythematous lesions. These are bright red, that is erythematous lesions with a central bluish or whitish dot. With a central bluish or whitish dot. They first appear opposite the lower second molars, all right, and they are pathognomonic of measles. And after the involvement, after they appear opposite the lower second molars, then they progress to involve the rest of the buccal mucosa, all right. So day 10, fever is followed by complex spots, which appear in turn 1 or 2 days before the appearance of the rash. Okay, before the appearance of the rash. Now, talking about the rash, guys, measles rash, very, very important. The rash of measles. First and foremost, you should know where this rash begins. It appears. The rash appears first behind the ears, all right. Then it comes on the face, then on the face, and then it spreads from top to bottom, including palms and soles. So this is how it appears, and guys, it disappears in the same order. The rash appears in this order and the rash disappears in the same order. And the rash is described as what? The measles rash is a maculopapular rash, all right, and it assumes a brownish hue in a few days, all right, and it may become confluent also. Confluent means the individual rash patches may become fused with each other, as if some reddish paint has been poured on the body, something like that. Okay, so that is called as confluent rash. So it can become confluent too. Now, guys, the rash, I said, disappears in the same order as in which it appears. Now, guys, have a look at this. Okay, so guys, here we have the model of the paramyxo. This is the model of the paramyxoviruses. Now, disclaimer, guys, enterotoxin is not to be confused with endotoxin. Remember, where have I talked about endotoxin in detail? Endotoxin I have discussed in detail in the bacterial cell anatomy or structure, and for your revision, let me remind you, it is an important or integral part of the outer membrane of gram-negative bacteria. Okay, so enterotoxin, please remember, it is not at all, not to be confused with endotoxin. Okay, enterotoxin, entero means gut. So basically, these toxins have got something to do with the gut. They are going to act on the intestines, and enterotoxins, first point you need to remember that these are exotoxins. Okay, exotoxin and endotoxin we have discussed in virulence factors in general micro. And if you remember, exotoxins are what? Proteins in nature, and proteins are heat labile. But the exquisite or the unique feature of enterotoxins is that even though they are a kind of exotoxins, yet they are exquisitely heat stable. How much heat stable they are? They can withstand a temperature of 100 degrees Celsius for 30 minutes or half an hour. This is a repeatedly asked question, all right. And the name of these toxins is enterotoxins. So what disease they are associated with? Remember, they cause food poisoning. Also remember, they are super antigens. Now, guys, have a look at this. Now, a very important feature I would want to tell you about Staph aureus food poisoning is the fact that, remember, even though Staph aureus is a pathogen, but in 20 to 40% of the population, Staph aureus is innocently present on the, in certain areas, simply as a colonizing organism. Now, the most common site of carriage of Staphylococcus aureus is anterior nares. And the second most common site of carriage is skin. Okay, remember this. Now, I personally look upon, Staphylococcus aureus, as I personally call it, notorious knock aureus. Knock means nose. All right. So imagine you go to a restaurant and the chef over there is a carrier of Staphylococcus aureus, harboring it in his anterior nares, and he has got the habit of nose picking, and he does not have the habit of washing his hands before he himself eats or he prepares the food. Okay. Now with those dirty nose picked hands, suppose he prepares the food and the staph aureus from his nose, they happen to enter the food, and on cooking, and suppose the bacteria now which have entered the food, and suppose they have already produced the enterotoxins, right? Now while cooking the food, the bacteria will be killed. The bacteria are heat labile, but the enterotoxins which have already been produced by the bacteria, which are exquisitely heat stable, they will not be destroyed even on cooking. So when such food will be served to you, what you are going to eat along with the food? You are going to welcome to your intestines not just the food but the enterotoxins also, and then you will have food poisoning. Okay. Similarly, if suppose there is some other chef, for instance, who does not have the habit of nose picking, but the skin of that chef is harboring staph aureus and he does not wash hands, again same story will be repeated. With dirty hands he's preparing the food, the bacteria are overactive, they have already produced the toxin, so on cooking the food, while preparing the food, the bacteria will be killed but not the toxins, and such food when served to somebody, that somebody will be eating not just the food alone but also the toxin. Okay, so this is how the toxin is going to enter the body. So note down, the most common site of carriage of Staphylococcus aureus is the anterior nares, and the second most common site of carriage is skin. All right. And remember the clinical features of Staphylococcus aureus food poisoning. Vomiting is a very typical feature. Diarrhea is rare. Okay, vomiting occurs because of vagal stimulation. Okay, vomiting occurs because of vagal stimulation. All right, and the types of enterotoxins of Staph aureus are A to G except F, A to G except F. And remember, this food poisoning is a self-limiting disease. You need not give any antibiotics because anyways it is not because of the bacteria, it is because of the toxins. Okay, even no antitoxin is indicated because in 3 to 5 days the patient will be normal. Okay, the patient will be fine. So it's a self-limiting disease. Okay, so remember all these important facts about Staphylococcus aureus food poisoning. And yes, one more thing, what is the incubation period, guys? The incubation period is 1 to 6 hours. It is such a short incubation period because this food poisoning is occurring because of the preformed toxin. Okay, there is no time required for the elaboration of toxin. The toxin itself is being eaten along with the food. We have got 4 entities and I'm gonna take up 2 and 2 together. Now first and second entities are known as Chirion and Favus. Alright. Now what is Chirion? It is an intensely inflammatory, boggy swelling of the scalp. It is characterized by scarring alopecia, and Favus is characterized by cup-shaped lesions called as scutula around the hair shaft. Okay, now you need to next know the causative agents of Chirion and Favus. Have a look at this, guys. Now, I on purpose wrote the names Chirion and Favus from left to right because I want you all to remember this. Okay, what you can see over here on the extreme left. Okay, this is KFC logo which I have drawn, and from KFC logo take home message is K, F respectively stand for Cheiron and Favus, and C stands for capitis. Alright, so remember, Cheiron and Favus are the types of Tinea capitis, and they are caused by what? Cheiron is caused by Trichophyton mentagrophytes and Trichophyton verrucosum, and Favus is caused by Trichophyton schonlini and Trichophyton violaceum. Alright, so the genus name is Trichophyton only, whether it is Cheiron or it is Favus. Now the names on purpose I have written in alphabetical order from left to right. Okay, so we have got from left to right M and S, V and V. Verrucosum and violaceum are also in alphabetical order. Okay, verrucosum comes before violaceum. So this is just a memorizing tip from my part. I hope it helps you. Now, Cheiron and Favus are two types of Tinea capitis, and the other two types I want you all to remember are ectothrix and endothrix. Alright. Now, leishmania causes the disease leishmaniasis. Alright, there are many species of genus Leishmania, and leishmaniasis, this organism has got animal reservoirs, So most of the types of leishmaniasis which occur because of different species, they are regarded as zoonotic diseases except one. Okay. Now let us see what is that important exception. See, as far as the species of genus Leishmania are concerned, the most important for us is Leishmania donovani. Alright, it causes visceral leishmaniasis, also known as Kala Azar. Kala Azar is an internationally accepted term. Alright, now Leishmania donovani is a strict human pathogen. So guys, this is an exceptional kind of leishmaniasis, is that visceral leishmaniasis is not a zoonotic disease. There is no animal reservoir for Leishmania donovani. It is not a therefore zoonotic disease. Okay, and these leishmaniasis is a vector borne disease. Alright, so the vector over here is sandfly, that is Phlebotomus argentips. It is called as, guys, argentips because the wings are silver in color. Okay, and remember, leishmaniasis can be prevalent in old world, called as old world leishmaniasis, old world meaning Asia, and new world leishmaniasis meaning the Americas, Central and South America. Vector for all kinds of leishmaniasis is sandfly. Okay, sandfly only. And far as the genera are concerned, do remember the two names Phlebotomus argentips for visceral leishmaniasis and Phlebotomus sargentii for cutaneous leishmaniasis. Generically also you should know, in case of old world, the the genus of sandfly which is prevalent is Phlebotomus, and in new world it is Lutzomyia. So let us see, if suppose this is a macrophage, this green circle represents a macrophage, and inside this is the nucleus of the macrophage. Now guys, inside this macrophage, let me draw one amastigote. See, this is an amastigote form. Okay, this is amastigote. This is approximately 2 to 3 microns. Okay, 2 to 3 microns in size. Now we have got this one, this is the nucleus of the amastigote form, and perpendicular to the nucleus, this orange dot which I have drawn, this orange dot represents what? This orange dot represents kinetoplast. Kinetoplast represents the mitochondria of the organism. It represents the mitochondria of the organism. Right, and guys, I have drawn just one amastigote. Macrophages are usually filled with amastigote forms, so they are teeming with or filled with amastigotes, and this whole structure, macrophage filled with amastigotes in this fashion, this whole structure is referred to as what? This whole thing is referred to as LD body, that is Leishmania donovani body. If we talk about Wuchereria bancrofti, figure A shows the schematic representation of microfilariae. Right, now see this. Now, in different filarial worms, we can differentiate them based on their morphology. Like in case of Wuchereria bancrofti, in this case, you can see a sheath, a hyaline sheath is there inside which the larva moves. All right. So, and this larva has got a column of nuclei. Now, as far as Wuchereria is concerned, you have to see the arrangement of the nuclei. All right. For instance, you have to notice the tail tip where no nuclei are seen. All right, no nuclei are seen. And look at this cephalic space, which is sort of empty. And on the right, you can see this is the microscopic view of the adult male worm. And at the left, and this is male worm, and this is female worm. Okay, very tiny worms they are. Varicella-Zoster virus, accepted short form is VZV. Now, this is one virus, and it causes two diseases, right? Two diseases, that is varicella and zoster. Both of these are names of diseases. Two diseases, varicella, also known as chickenpox. Varicella, also known as chickenpox, and herpes zoster or simply zoster. So zoster or herpes zoster, also known as shingles. All right. Now, guys, some basic points about these diseases. Okay. Before that, how does this virus, how does the infection occur? Now, remember, in case of Varicella-Zoster virus, the infection occurs by droplet infection and contact with the skin lesions. The skin lesions are also infectious. All right. Now, guys, as far as these two diseases are concerned, in the the very first video on herpes viruses, that is herpes virus part 1, before I started talking about HSV1 and HSV2, I told about some general facts about herpes viruses. That is, very first time a herpes virus causes infection, it is called as primary infection. Then it becomes latent or hidden in the body at some site or the other. Then later on, it can wake up from sleep and cause reactivation infection. Okay, so applying the same concept over here, as far as this herpes virus, Varicella-Zoster virus is concerned, varicella or chickenpox is the manifestation of primary infection. So this is the manifestation of primary infection. Whenever this virus infects anyone, whether it is a child or an adult, varicella or chickenpox is going to be caused. All right. Now, having said that, let me also say that chickenpox is a childhood disease. Actually, it's very common in children. Most commonly, it occurs in children. So, it is a childhood disease. Childhood disease. Remember, C for chickenpox, C for childhood. But what happens in old age, especially in the 6th decade of life, the antibodies either become totally absent or they become decreased in number, and this predisposes the individual to develop reactivation infection. Okay, so that reactivation infection is nothing but zoster. Okay, so zoster or herpes zoster or shingles is the manifestation of reactivation infection. Okay, it is common in the 6th decade of life. It is characterized by vesicular rash, vesicular eruption along the course of a sensory nerve of head or
diseases. Recommendations for VZIC Varicella-Zoster immunoglobulin administration. Exposure criteria. देखो ये बढ़िया सा टेबल है, देख लो इसको. Exposure to person with chickenpox or zoster. ठीक है? If we are exposed to somebody with zoster, then if we don't have antibodies, then also we are susceptible to infection. Not just if we are exposed to a case of chickenpox. अब देखो household. ये pregnant lady की niece जो थी वो दोनों they were staying in the same house. ठीक है, household residents in the same household. ठीक है. Playmate, face to face indoor play. Hospital, varicella, same 2 to 4 bedroom or adjacent beds in large ward, face to face contact with infectious staff member or a patient, visit by a person deemed contagious. Zoster के case में intimate contact like touching, hugging with a person deemed contagious. Newborn infant, onset of varicella in the mother less than or 5 days before delivery or less than 48 hours or equal to 48 hours after delivery. Okay, VZIG, Varicella-Zoster immunoglobulin is not indicated if the mother has zoster. ठीक है, ये हैरिसन की गाइडलाइंस है. Patient should receive Varicella-Zoster immunoglobulin as soon as possible but not more than 96 hours. तो ये तो हमने एक्सपोजर क्राइटेरिया डिफाइन कर दिया कि ये बंदे एक्सपोज्ड होंगे. अब ये जब समझ में आ गया कि ये व्यक्ति एक्सपोज है तो करोगे क्या उसके लिए. ओके, I mean these are the candidates actually. So these are the criteria and these are the candidates. Immunocompromised people, immunocompromised, susceptible children without a history of varicella or varicella immunization, susceptible pregnant women, newborn infants whose mother had onset of chickenpox within 5 days before or within 48 hours after delivery, hospitalized premature infant whose mummy lacks a reliable history of chickenpox or serological evidence of protection against varicella. मतलब कि मम्मी के पास सीरम में एंटीबॉडीज नहीं है, or the test for antibodies could not be done in the mother. Hospitalized premature infant, regardless of maternal history of varicella or varicella-zoster virus serological status. ठीक है, तो fifth point का मतलब ये है कि when you don't know the status of the mother, better to take, better to give a benefit of the doubt to the infant and give varicella-zoster immunoglobulin. So this table is very important. There's an outbreak of encephalitis in a community. According to universal immunization schedule, the route of administration of the vaccine for the likely infection is, पहली बात तो outbreak of encephalitis in a community. What is the most common cause of epidemic encephalitis? ये मैंने again NEET PG में ही QRP में भी cover किया था. It is Japanese encephalitis. तो उसकी वैक्सीन के बारे में पूछ रहे हैं. Okay. We give live subcutaneous in the under the universal immunization schedule. Killed subcutaneous नहीं होती. Killed, remember KIM. ठीक है, KIM से याद कर लो. K for killed, I am for intramuscular. I am for intramuscular. ठीक है, तो live subcutaneous है आंसर इसका. बाकी तो option could not be recalled. I did not get. If you get, please inbox me.
Number 2, most common cause of lobar pneumonia. Number 3, most common cause of acute meningitis in adults. The seventh organism in Enterobacteriaceae family is Yersinia pestis, which causes the disease. It causes the disease plague. Yersinia pestis again is kind of a lazy organism in the sense that it is NM, that is non-motile. Okay, it is non-lactose fermenter. It is also non-hemolytic. Now the characteristic kind of growth produced on oil or ghee broth is described as stalactite growth. Stalactite growth. Now have a look at this, guys. In this picture on the left you see the actual stalactites. Stalactites are these hanging things projecting downwards from the ceiling of caves, all right, like this. And somewhat similar to these stalactites, the growth is produced by Yersinia pestis in culture media. This is oil, this is a culture medium, liquid culture medium, at the top of which oil or ghee is floating, and from that you can see these downward, inverted, this kind of morphology is described as stalactite like growth of Yersinia pestis. Now another factor which you need to remember regarding the growth characteristics of this organism is that it is non-fussy, that is it is non-fastidious, so it can grow on nutrient agar, nutrient broth. And in fact the stalactite growth which I was referring to, in this we take nutrient broth only, but we at the top of it we put a layer of oil or ghee, so it becomes oil or ghee broth. Okay, So let's say this is the oil or ghee layer, and this is the nutrient broth. So it is non-fastidious, and if it is non-fastidious and you give it an enriched medium to grow, it will grow on enriched media also. That is, on blood agar also, of course, it will grow, and on blood agar we can also comment on hemolysis. Is it hemolytic or non-hemolytic? Remember, it is a lazy fellow. It is non-hemolytic. Okay, and on blood agar, brown colonies form because of absorption of hemin. All right, now even though we say it is okay, it is Enterobacteriaceae family, so it has to be gram negative rod, but guys, this is not a proper or a perfect rod like this pen is. It's actually described as a cocobacillus. Okay, it's a gram negative cocobacillus. It is neither coccus, that is, it is neither perfectly spherical, nor it is a perfect rod or bacillus like this. It is somewhat intermediate between a sphere and a rod, so somewhat like this, meaning oval looking organism. So gram negative cocobacillus. And remember, in general micro also, I have mentioned during the discussion on capsule, the only two bacteria in the entire bacteriology which have got protein capsules are Bacillus anthracis and Yersinia pestis. So it is a capsulated organism. All right, the capsule has F1 antigen. This F1 antigen is plasmid encoded. The capsule is antiphagocytic. And it is produced optimally at a temperature of 37 degrees Celsius. It is produced optimally at 37 degrees Celsius, that is at human body temperature. All right, but the optimum temperature for growth of Yersinia pestis is actually 27 degrees Celsius. So the optimum growth temperature for the organism is 27 degrees Celsius, but the optimum temperature for capsule production is 37 degrees Celsius. Okay, now talking about the important virulence factors. Remember, one is the F1 antigen, which is present in the capsule. The capsule itself is a virulence factor because it is antiphagocytic. Then Yersinia outer membrane proteins, that is OMPs, Yersinia outer membrane proteins. Then LPS, that is lipopolysaccharide. Remember, it's a villain it's a weapon. It's a villain for us and it's a weapon for the gram negative organisms, being present in the outer membrane. All right. Then siderophores. Siderophores help in the acquisition of iron. Okay, siderophores help the organism in acquiring iron. This iron then can be used in the metabolic pathways used by the bacteria. All right, now the disease which this organism causes is known as plague. Okay, it is a zoonotic disease, that is, it is acquired from animals. Right, now the reservoir, now since it is a zoonotic disease, expect the organism to have some animal reservoir. The reservoirs are actually wild rodents. For example, field mice, bandicoot, etc., and not the domestic rodent Rattus which was initially thought. Okay, and the vector is rat flea. Xenopsilla chiopus. All right, the flea is going to feed on the rodents, and the most common mode of infection in humans for this disease is bite of the infected rat flea. That is the most common mode of infection. So modes of infection. Number 1, most common, MC means most common mode is bite of the infected rat flea. Then some other modes are also there, for instance, contact with the tissues of the infected rodents. Okay, then inhalation also, in case of what? Pneumonic plague. A patient of pneumonic plague is infectious. The person can pass on the infection to other people. The mode of infection is inhalation. I already have mentioned this important point that pneumonic plague is the only exceptional zoonotic disease which is transmitted from man to man. Okay, this I have discussed while the discussion of Chlamydophila psittaci. Okay, please check that out. And so this is the notable exception which you need to remember. And very rarely, due to contact with human flea, this is another mode of infection, although rare, bite by human flea, that is Pulex irritans. Pulex irritans.